So next to passive transport, we have active transport. In active transport, mainly the active transport, it is categorized into three major categories or three major groups, right? So first I have given here children under active transport, Roman number 2, which I have given. Under this we have three categories to be discussed. The first one I have numbered it as 1, which is primary active transport, and second one I have given it here children, you can see this, right? So this is 2, which is secondary active transport, and the third one we have group translocation. So all these three will come under the active transport. So what are the three categories we have? Primary active transport, we have secondary active transport, and also the last one being the group translocation. So all these three categories will be discussing shortly. So before going to discuss with these categories, first we have to be aware of this active transport. So what is this active transport? What type of process is this? How the movement of the molecules will be taking place, and what is the basis for this active transport? So we'll be learning the important features of the active transport system. Okay, so here as we have discussed, discussed with the passive transport features, in the same way we'll be learning with the active transport features here. So mainly the important features will be writing it down, right? So if you remember, in the case of passive transport, in the introduction of the transport, I have told you that this process is said to be osmosis. What is osmosis? Osmosis, it is defined as the movement of molecules from higher concentration to lower concentration through a biological membrane or through a membrane. It is known as osmosis. It is a physiological process that I have discussed before. And if you compare this thing with the active transport, can you call this active transport as the osmosis? Think of it children. Osmosis means it is the movement from higher to lower concentration. Then what about the active transport? I have told you that active transport, it is against the gradient. It is not at all moving along the gradient, it is moving against the gradient. Here the molecules will tend to move from lower concentration to higher concentration, which is a reverse of the normal situation or the normal state, that is the from higher concentration to lower concentration. Generally the molecules will tend to move from higher to lower, which we call it as osmosis if it is happening through a membrane. But in the case of active transport, the molecules move from the lower to higher concentration. Then can you call it as osmosis? We cannot call it as osmosis, but we call it as reverse osmosis. Okay, so the opposite of osmosis is happening here, and hence we can call this movement as reverse osmosis. Very important, right? So it is, are you getting this children? Reverse osmosis. Why? Because the movement of molecule, it is happening from lower to higher concentration through a membrane. Hence we call it as reverse osmosis. Even in the purification of water, we use this technique, that is RO water. Generally we call it as RO water. RO water, it is nothing but the major important technique that is employed as the reverse osmosis technique. Okay, in absorption of the waste substances or impurities or salts, that will mainly take place with the help of a membrane through reverse osmosis technique. Okay, so the same thing it is applied here in active transport when we have this reverse osmosis. This is the first important point. And second important point, the features we will be writing now, that is uphill movement, right? So in the case of the passive transport, what I have mentioned children, it's a downhill movement. Why? Because the movement is from higher to lower, it is called as downhill, and here it is from lower to higher, which is called as uphill movement, just like climbing the steps, right? What about the gradient? Gradient is definitely present, but the gradient, it is the molecules are moving not along the gradient, they are against the gradient here. Okay, so they are against the gradient. Okay, so clear with this point? It is against the gradient, it is uphill movement, it is called as reverse osmosis, and this is happening from higher to low concentration. Okay, then what about the other important thing here we have to discuss regarding this? As it is uphill movement, it is simply like climbing steps. I have told you it requires more of energy. So as it is energy expensive process, all these actively, or you can say wherever this active transport it is more in the cells or the organelles, there you can see a lot of mitochondria. Why? Because we all know mitochondria is a power supply. It will be supplying the ATP, which is the energy currency. So wherever the active transport it is mainly taking place, there the cells will be showing the large amount of mitochondria. Okay, so mitochondria are mainly associated with the cells having this active transport. And in this active transport, the important thing here we have to discuss is regarding the energy. Energy, it is essentially present for each and every molecule to be passed in or out. I have told you, even in the passive diffusion or passive transport, the energy requirement is there. For a molecule to be moved in or out, it requires certain energy. But in the case of passive transport, this energy, it is the inbuilt energy, or you can say each and every chemical molecule, they have some innate potential energy. While moving, they will create the kinetic energy, and this energy will become the driving source for their movement. But in the case of the active transport, as they are building this gradient, which is the uphill movement, it is against the gradient, right? The molecules are not moving from higher to lower, it is moving from lower to higher. Then it requires the additional energy, and this additional energy, it is sourced from the cell's energy currency, that is nothing but the ATP. Okay, so we have only one source present inside our body or any animal. If you take the energy currency is the ATP, and these ATP molecules will be present inside the cell, and these molecules are used for pushing these molecules against the concentration gradient. Okay, so here in the case of active transport, the ATP is very, very essential or mandatory. So here again, in certain cases we have some transport systems in the active transport where the ATP is not required. The energy will be required for each and every molecule, and it is supplied to the system differently. In passive transport, they will be having their inbuilt potential energy, or you can say the molecules will be using the entropy or the kinetic energy present in these molecules, which are naturally present. Okay, for active transport, they require additional energy, and this additional energy will be supplied from the ATP, which is present inside the cell cytoplasm. Okay, and in this active transport again we have the second case. What is the second case? Certain systems or certain proteins which are helping in this transport, they are not utilizing this ATP directly, rather they will be utilizing these concentration gradients created by other molecules. Are you getting me, children? I am saying you that the energy is required. In active transport, we have two conditions or two situations. The energy is definitely required. Some molecules, they are very faithful and they will use the direct energy, that is ATP. Okay, so they will depend upon the cellular energy, which is the ATP. And there are certain molecules, they will take advantage of other molecules. There are certain other molecules which are moving along their gradients. Whenever they are moving along their gradients, they will have certain energy, and their movement will be releasing some entropy, that is the thermal energy which is released is also used here. So kinetic energy or thermal energy which is released by other molecules, it will be stolen by molecules which are present in the active transport. Okay, so here what is happening, what I am trying to say is, in active transport we have two conditions. One, there are certain molecules which will be utilizing the ATP directly without any hesitation. They will be borrowing the cellular energy, which is the ATP. There are certain proteins which are present in active transport. They will be borrowing this energy from different molecules which are moving along their chemical gradients. Okay, so whenever there is the chemical gradient, when they are moving along the gradient or downhill, they will definitely definitely have certain amount of potential energy or entropy that is released, and this will be stolen or it will be borrowed from them, and they will take advantage of other molecules and they will be utilizing in their own movement. Okay, so this is how active transport is mainly operated. Okay, so let us see the second condition here. ATP is required for the active transport. So what is the first point I'm writing here? Transport, the active transport requires ATP. Okay, and second important point, the transport in the active transport system even utilizes the chemical gradients of other molecules, okay, which will help in their own movement. So look at this carefully, children. So what I'm trying to say here is the transport. In active transport, we have two situations. Active transport can occur with the help of ATP. Why? Because they are moving against the gradient, so they require additional energy, and this additional energy can be sourced either from ATP, which is directly available inside the cell, or it can be borrowed from the chemical gradients of other molecules. The other molecules will have the energy while moving downhill. They will release certain amount of energy, and this energy will be borrowed or shared between between the molecules participating in the active transport. We'll be discussing shortly how these chemical gradients are helpful in the active transport. Okay, so simply remember, the transport can occur with the help of ATP or with the help of the additional energy sourced from the chemical gradients of other molecules. Okay, so this is what you have to remember here. And the transport happening, the active transport which is taking place with the help of ATP, it is known as the primary active transport. So what is primary active transport? Is it clear, children? So the transport which is taking help of ATP directly for its movement, we call it as primary active transport. Then what about the transport which will be utilizing the chemical gradients of other molecules? It is known as secondary active transport. Clear with this? Primary active transport is the one where the ATP is required directly, and in the active transport where the ATP requirement it is not present, but energy will be sourced from the chemical gradients of other molecules. We call it as secondary active transport. So both are part of active transport. Remember, why? Because they require additional energy to move or push the molecules against their concentration gradient, and hence these are categorized under active transport. But what is the difference? In primary active transport, the ATP, it is directly utilized, or ATP is mainly seen in the case of primary active transport, but in the case of secondary active transport, ATP is not present. So ATP is not the source of energy, but the source of energy is different, which is the chemical gradients of other molecules which are moving downhill. There are certain molecules which will move downhill, which will help in sharing this energy with other molecules. Okay, so that is regarding the secondary active transport. So these are the important requirements, or you can say the basic features of the active transport. And coming to the proteins which are participating in this active transport. So in passive transport, what are the proteins we have discussed with children? Remember, I have discussed with the first important protein, the channel protein. The channels are again non-gated channel, gated channels we have discussed, and another type of protein we have discussed, the carrier protein. So there are two important proteins in the passive transport. One is the channel protein and other is the carrier protein, and we have seen how these structures are mainly present. These are transmembrane proteins embedded inside the plasma membrane, and one is like the pore creating channel. And another important thing I have discussed in the passive transport, that is regarding carrier protein. The carrier proteins will mainly show the conformational changes, right? So these two will come under the facilitated diffusion, right? So in this, the carrier proteins, they doesn't create any pore in the membrane, but simply they will have the site of attachment like the revolving gate. In the same manner, the carrier protein will be having the similar structure like that. So one molecule, it will be present in above. When it is attached to it, later it will be changing its conformation and it is released inside the cell cytoplasm that we have discussed before. So here in this carrier protein, if you see, the conformational changes are mainly present. And the most important thing I have discussed is the importance of carrier protein in both passive as well as active transport. Carrier proteins are common for both passive as well as active transport system, and these carrier proteins will be having the same functions. So whatever functions which I have discussed with the carrier proteins in the passive transport are same with the active transport as well, right? So what are the important functions of the carrier proteins? I have told you, one, they have the conformational changes, and second, I have discussed with the site of attachment. Okay, and one important difference we have in the carrier proteins of passive as well as active transport, that we are going to discuss now. So the carrier protein in the active transport, mainly it will have many sites of attachment. This is the one difference you have to remember. Other common things what I have told you, one, they will show the conformational changes just like the carrier protein of the passive transport. Here even we have the carrier protein. So this is the carrier protein picture which I have given for the active transport. If you observe carefully, there is a slight difference. In this case, we have only one site of attachment. If you closely observe the diagram, here I have given one site of attachment for the carrier protein in the passive transport. But what about the carrier protein, which I have mentioned as active carrier protein? Here you have two sites. I have mentioned two sites, children. Look at this. This is one I have marked, one molecule it is marked in red and other molecule in blue. So look at the structure carefully. So this is the carrier protein which is participating in the passive transport, and this is a carrier protein participating in the active transport. So in this molecule, if you observe, this is one site of attachment where the substrate will be attached. Okay, so this is the substrate attachment site. Any molecule, it can be some glucose or any ion, any molecule can be attached here. But here, as it is active transport, the ATP is required. So there must be some site of attachment for ATP even. Okay, so this is the site of attachment for ATP. Okay, so this is for ATP. This molecule represents the adenosine triphosphate here. What will happen in this region, children? So in this region, along with the attachment of molecule, we have the attachment of energy currency, that is ATP. And immediately there is change in conformation. This molecule will be inverted like this and it will release the substrate inside. And all this will be happening with the help of the hydrolysis of ATP. One energy molecule is required to push one molecule into the cell or out of the cell. Okay, so here how the hydrolysis is happening. One ATP, it is hydrolyzed into adenosine diphosphate, ADP plus PI, and here immediately when the hydrolysis it is happened, the molecule will be inverted like this. So this is the upright conformation, and later it is inverted conformation and inverted configuration. In the same way, even this carrier protein, it is upright. After the hydrolysis of ATP, it will be inverted in this way. Okay, I have not given the inverted structure here in order to adjust the space. So simply understand the inverted structure will look like this. But here we have two sites of attachment. One is for ATP and other is for the substrate molecule. So once it is inverted like this, what will happen? The substrate is ultimately released, and even this adenosine diphosphate, when the ATP is hydrolyzed, the ADP and PI are formed. Immediately this ADP will be released or disassembled from the carrier protein, which will lead to the reorganization of this carrier protein back to its state. Okay, so once the hydrolysis it is done, it is a signal for the carrier protein to get back to its native state. It will be remodeling or reshaping itself, and it will be reverting back to its original state. So this is how mainly this carrier protein will be working here. Everything is same, the conformational changes, or you can say the site of attachments and the shape, everything will be same here. But the thing you have to remember, mainly they have different sites of attachment for substrates and also the ATP molecule. Here I am discussing with only one substrate. For this carrier proteins, we have more than one substrate. In the case of active transport, at a time we can see the movement of more than one substrate. Okay, so this is also a key feature for the active transport system. So carrier proteins are present in both. So all the transport systems which are operated under active transport, they mainly have these proteins, and these proteins are called carrier proteins. And how are these carrier? The carriers are same like that of the carrier proteins in the passive transport, but the major difference is the site of attachment. And here the site of attachment will be more for the carrier proteins in the active transport compared to passive. In passive we have only one site or specific site of attachment. These are highly specific molecules and they have only one site of attachment. Here even these are specific molecules, but the site of attachments will be more. And what I have discussed first with the substrate molecule, and another site of attachment is for the ATP molecule. So wherever ATP is required, they will be having the site of attachment for ATP. Okay, and another important thing which I am going to discuss is regarding the substrate molecule. So here more than one substrate can be attached to this carrier protein. So look at this. This is one substrate. Let us say some glucose. Along with glucose we have this another molecule which is moving along the carrier protein at a time, and such transport is called as co-transport. It is known as co-transport. Why? Because two molecules or three molecules, they are traveling together. We call it as the co-transport here. So such type of movement is also possible. So I will be discussing with these transport system with the examples which I have given here, children. Simply understand that the carrier proteins will show the conformational changes. They will have the site of attachment. Their working and functioning, it is same like that of the carriers which we have discussed before in the passive transport. And the most important thing here we have only one site of attachment for one substrate, but here we have multiple site of attachments, and one such attachment is for ATP I have discussed, and substrate I have discussed. Substrate molecules along with the ATP, we have different kind of substrates are moving along this carrier protein that we will be discussing here. Okay, so this is regarding the general features of the carrier proteins which are present in the active transport. And another important point here I will be writing here. So the carrier proteins, so what is a major difference for the carrier protein in active transport? The site of attachment, multiple site of attachment will be there. Okay, so that I am mentioning here. So here we have the multiple site of attachments are seen that you have to remember. So what are the basic features here you have to remember, children? You have to by heart all these features in order to understand the different categories under active transport. It is very important to be thorough with all these points. One, it is the reverse osmosis. I have told you. In the case of passive transport, it is osmosis, and active we have reverse osmosis. In passive we have the downhill movement, and in the active transport we have uphill movement. In the case of the passive transport, we have along the gradient which is from low to high. In the case of active transport, we have against the gradient, that is from high to low. And another important thing, in the case of passive transport, there is no requirement of ATP. That is additional energy, it is not required. The basic chemical gradient will be driving the molecules in and out. In the case of active transport, this is different, children. Why? Because it requires additional energy, but the source will be different here. Some type of active transport systems, they are operated with the help of ATP, and some of the systems will be operated with the chemical gradients of other molecules which are moving downhill while releasing certain amount of entropy, or you can say the kinetic energy. And this is regarding the active transport. So based upon the source of energy, it is again divided into primary active transport where ATP is directly required, and secondary active transport where ATP it is not directly required, but they will utilize the energy of the other molecules chemical gradients. This is what we have discussed. The last important point here is regarding the protein which is involved in active transport. Basically, the one protein which is functioning for this active transport is the carrier protein. So all the proteins present in the active transport will come under these carrier proteins. All are carrier proteins, and these carrier proteins means they have their conformational changes. They will be having, that doesn't form any channel in the membrane, they simply show the conformational changes. They will change their conformation with the attachment of the substrate. And most important thing which I have discussed here is in passive transport, the carrier proteins will have one site of attachment for a specific substrate, but in the case of the carrier proteins participating in the active transport, they will be having multiple sites of attachment. Why? Because here you can see the substrate molecules which are moving along with some helper molecules. I have told you there are certain cases where the help of other molecule is required. So generally these carrier proteins, they must be allowing some other molecules. So there are multiple site of attachment, and not only that, some of the systems they are operated with the help of ATP. So ATP is also has to be attached to the carrier protein. So there must be some specific site of attachment for them. All these sites, multiple site of attachments are seen in the carrier proteins of the active transport. So this is regarding the key features of the active transport. Now let us move on to the classification under active transport. We'll be discussing with the primary active, secondary active, and the third one is a group translocation. I will be discussing with the differences here along with the examples. So look at this carefully, children. So in active transport, first we will be discussing with the primary active transport. And what I have told you, you have to keep it in mind that all the proteins participating in the primary active transport are the carrier proteins. Okay. So here mainly with the help of the proteins only the movement of molecules will be taking place. Even for the passive transport, the molecules are moving along the concentration gradient with the help of the proteins here, and hence we call it as facilitated diffusion. Facilitated, why? Because they are supported with the proteins, and hence they are called facilitated. Only with the simple diffusion you cannot see any protein. So protein is absent here. In simple diffusion we don't have any protein. It is not required. Through some pores the molecules will be easily passing through this membrane. That is called simple diffusion. Okay. So this is regarding the passive transport in comparison with the active transport. Now we are starting with the first type, that is the primary active transport. So in this case, so the primary active transport, so let us write a classification here. So these are the three categories under active transport, children: primary active transport, secondary active transport, and group translocation. Primary active transport, once again I am telling you, ATP is directly required. Secondary active transport, ATP it is not directly required, but it will utilize the energy which is released by the chemical gradients of other molecules. This is regarding secondary active transport. So under this we will be explaining with each and every example which I have given. So let us take with the primary active transport. And what I have told you, all the proteins which are mainly participating are the carriers. They will show conformational changes in the release of the substrate molecules. And most importantly, these primary active transport carrier proteins are renamed as pumps. What it is known as? It is very easy to understand, children. I am telling you that the primary active transport carrier proteins, all are carrier proteins, but each will be named differently because of their functioning. The carrier proteins which are participating in the primary active transport, they are said to be the pumps, or these are also known as ATPases. So what is the name given to the carrier protein in the primary active transport? The carrier proteins are simply said to be pumps or ATPases. Why? Because they are utilizing this ATP to push the molecule against the gradient, and hence we call them as pumps. They are very powerful. Why? Because they have extensive energy. The energy system, it is operated here. The high amount of energy, it is utilized to pump the molecules, and hence we call it as pumps. As they are utilizing ATP, these enzymes which are hydrolyzing ATP are mainly associated with these proteins, and hence we call it as the ATPases. So wherever you see the ATPases or pumps, you must understand that these proteins are related to the primary active transport. This is very, very important regarding your NEET children. So primary active transport, it is mainly associated with the which type of carrier proteins? These carrier proteins are known as pumps or ATPases. Why? Because they will be utilizing the ATP to pump the molecules against their gradients. Okay, so this is regarding primary active transport. Then what about the proteins which are present in the secondary active transport? In secondary active transport, the proteins are simply said to be the carrier proteins, but here mainly these carrier proteins will allow multiple molecules to move together. Okay, so here what will happen? The molecule movement will be the substrate which we have discussed before. Some of the carrier proteins will have multiple sites of attachment where 2 or 3 substrates will be moving together, and such type of transport, it is said to be the co-transport, which I have discussed before. So the co-transport system, it is mainly seen in the case of secondary active transport, and here the carrier proteins are renamed as co-transporters. Okay, so here in the secondary active transport, the carrier proteins are mainly of which type? Co-transporters. All these molecules are co-transporters. Exclusively the carrier proteins are completely the co-transporters in the secondary active transport system. Why? Because here the molecules, the movement of one molecule has to be supported by other molecule, which I have told you before. The molecule will utilize the gradient of another molecule, so this molecule is directly linked or coupled to the primary molecule, and hence both the molecules will be coupled and they will move together. So this is exclusively the co-transport system. Why? Because the molecules are in association, and this is regarding the secondary active transport. Then what about group translocation, children? Group translocation is also requiring this ATP, but here mainly the carrier proteins are multiple subunit of enzymes here. Okay, so not the single unit of protein. You can see many proteins are associated with the enzymes, and hence we call such carrier proteins as multi-subunit complexes. Okay, so here you can see. So this is regarding each transport system and their proteins. Okay, so all these are carrier proteins. Carrier proteins which will take the help of ATP are renamed as pumps. Carrier proteins which are mainly associated with more than one molecule, we call them as co-transporters. Carrier proteins which are associated with many enzymes, proteins, forming a complex protein subunit for entry of molecules, we call it as multi-subunit enzyme complexes. So all these three are present for the active transport system. All are carrier proteins. Why we are calling it as carrier protein? Because they are showing the conformational changes, and hence we call them simply as the carrier proteins. But based upon their functions, they are named differently. And another important point here, so I have told you here the pumps are operated. Even here the single or two, you can say the single or multiple molecules can even pass in the primary active transport. So the co-transporters are also seen in the case of primary active system. Along with pumps, we have co-transporters. So both are seen. Pumps are seen, pumps. Along with the pumps, we have the co-transporter. So two type of proteins will be seen, that I will be clearly explaining each and every concept. When I am discussing with all those concepts, then it will be very easy for you to understand this classification. Okay, so let us move on to the classification. The first one being the primary active transport. So in this primary active transport, the example which I have taken today to explain you regarding the concept is the sodium potassium pump. Okay, so let us see how the sodium potassium pump will work. So the sodium potassium pump, it is also known as sodium potassium ATPs. Okay, so why it is known as ATPs? Because mainly the pumps are operated based upon the adenosine triphosphate, that is ATP, and hence it is also known as the sodium potassium ATPs. Now tell me, how many of the molecules are participating here? So for this example, if you observe, the sodium along with the potassium, both are obligatory, means each molecule requires the support of another molecule. The sodium, it is mainly creating the gradient and it will help the movement of the potassium ion. Okay, so both are coupled here. So such type of transport where more than one molecule is participated, we call it as a co-transport. Okay, and the protein involved in this co-transport, it is called as the co-transporter. As I told you before, the system which is operating for this co-transport, we call it as the co-transporter protein molecule. Now let us see how the sodium potassium pump will work. The sodium potassium pump, it is present on all the membranes. It is universal and it is present in all the membranes of the animals without any exception. This is very important protein molecule. Why? Because this protein molecule, it is mainly maintaining the concentration of the sodium extracellularly. Then why it is important to maintain the sodium concentration in the extracellular region? So that we will discuss now. First we will see the significance. So look at this, inactive primary active transport. The example which I am discussing now is the sodium potassium pump. Okay, so significance here, mainly what is the function of the sodium potassium pump? It will maintain the sodium ion concentration in the extracellular region. Not only that, it will maintain the potassium ion concentration inside the cytoplasm. Then why it is important for the sodium ion to be maintained in the extracellular region? That we will discuss. The major ions that are mainly present in the cell cytoplasm are the sodium ions, and the sodium ions will play an essential role in creating the gradients. As I told you before, this co-transport system, it is operated with the gradients of other molecules which are moving downhill, and in this mainly the sodium ions will play a major role. Along with the sodium ions, we have hydrogen ions. Both these ions will play a major role in creating the gradients, and these gradients in turn are helpful in the co-transport movement. Okay, so here there are two main functions. One is this sodium potassium pump. It will be maintaining the sodium ion concentration in the extracellular region, and it maintains the potassium ion concentration inside the cytoplasm. Now I will tell you why it is important to maintain the sodium ion concentration in the extracellular region. One, you have to remember, the glucose is the most important molecule inside our body or inside any living organism. Why? Because this is an easy absorbable form of energy. The instant energy is also derived from the glucose, and this glucose is constantly produced inside the cells, and it will be absorbed into the cell cytoplasm. But in order to absorb the glucose, there are certain challenges that are faced by the cells. One, the size of the glucose molecule, it is very large and it is highly polar molecule. As it is polar in nature, it is mainly reacting with water, and it is creating hydrogen bonds with water. Just if you observe for the channel protein, I have told you the water molecules can easily enter, but even the glucose, as it is the common form of energy, the cell can adapt a system where a glucose can easily be entered. But what is the main challenge or major challenge? The size of the glucose. The size is large, and second is the chemical nature. The chemical nature being it is highly polar in nature, and hence it can't be easily or passively transported into the cell system. And not only that, the glucose, if you observe, the glucose, it is traveling along the plasma membrane in different conditions. Sometimes even the concentration it is low inside, it has to be traveled against the concentration gradient due to various conditions, and all these are supported with the help of the co-transport. Okay. Mainly the glucose entry, it will be supported in the cell cytoplasm with the sodium ion gradient, or you can say the sodium ions are mainly helping in the transport of glucose molecules. Not only glucose, children, if the sodium ion concentration it is maintained extracellularly, if it is more, it can transport through its gradient, it can transport glucose, the more commonly used metabolites like amino acids and some important sugars. All these molecules, they require energy, and if energy, it is every time if the cell it is utilizing its cellular energy or ATP, it will be very expensive for the cell, and it is a wasteful process. So what it will do, it will be targeting the ions which are mainly excessively present in the extracellular region, mainly the sodium ions or the protons, and these ions, they will be moving downhill, and the energy will be shared with these molecules, and that energy in turn will be utilized by the substances like glucose, amino acids and other substances which are very important and constantly exchanged between the cells, and they will be entering easily into the cells. Okay. One, they doesn't have any easy carriers. So what is the second way the cell is adapting? It is mainly depending upon the gradients of other molecules which are excessively present in the cell cytoplasm, or you can say in the extracellular region. So what are the substances which are mainly present? The ions, and in ions again the sodium ions and the hydrogen ions, they are mainly essential. They has to be maintained in higher concentration in the extracellular region, so that the movement of such molecules from higher to lower concentration downhill, it will be releasing a lot of energy, and this energy will be utilized by different molecules which has to be constantly entering into the cell or leaving out of the cell, like glucose, amino acids, etc. Okay, so this is how the cell is mainly facing the challenge of entry of the glucose. So in order to maintain the sodium ion concentration, mainly the sodium potassium pump is working. Another important thing, it also maintains the membrane potential. We all know the cell cytoplasm, it is slightly more negative compared to the extracellular region, and this membrane potential is constantly monitored by the sodium potassium pump. So by the uptake, or you can say intake of this potassium ions inside the cell, so always the potassium ion concentration has to be more inside the cell. We are not going to discuss all these details. We'll be discussing this in the membrane potential while discussing with the nerve cells. I will be discussing all these things, children. So potassium ion concentration, it has to be maintained inside the cytoplasm, and sodium ion concentration, it has to be maintained extracellularly. So these points you have to remember simply. And in order to maintain this balance, the sodium potassium pump is mainly working. This is operating on all the membranes, and it is essentially present in all the living system, especially if you take animals. All the animal cells, they will be having the sodium potassium pump. This will create a gradient, this will create a balance of the sodium ions in the extracellular region, which makes other molecules to pass the membrane very easily. Okay, now let us see the functioning of the sodium potassium pump. So here, coming to the main concept that we have to discuss here regarding the primary active transport. Why we are explaining the sodium potassium pump under primary active transport, that we have to see. One, I have told you the primary active transport system, it will be operated in the presence of ATP. So now tell me, this is a co-transport system. Why? Because two molecules are operated at a time. And in this again, it is a primary active transport, so ATP will be generally required here. So for the movement of sodium ions outside the cell and potassium ions into the cell, there will be the energy molecule, which is ATP. Okay, so here we require, so ATP will be required here. Hydrolysis of ATP is essential for the operation of the sodium potassium pump. And the second important point, how many sodium ions are exchanged with the potassium ions, that is also important here. So in this case, if you observe, the sodium ions are transported, or you can say exported outside, exported out of the cell, and the potassium ions are imported into the cell. But every time when it takes the potassium ion, how many potassium ions are mainly taken in? For every time a pump operates, the sodium potassium pump operates, it will be mainly associated with 3 sodium ions on one side and 2 potassium ions on other side. Okay, which means if 3 sodium ions are let out of the cell, let us say 3 sodium ions are exported out of the cell, in turn, in exchange, we have the 2 potassium ions which will be entering into the cell. Okay, so this is import and this is export, or vice versa can happen based upon the needs of the cell. This can also be reversed, but it is always in opposite direction. Never potassium and sodium, they will be moving in the same direction. It is always in the opposite direction. As they are operated, as this co-transporter system, it is operated in opposite direction. We call such system as antiporter. If 2 molecules are operated in the opposite direction, we call it as a antiporter system. Okay, why it is antiporter, children? Because 2 molecules are operated here and these molecules are operated in the opposite direction. So in exchange for 3 sodium ions, 2 potassium ions will be entering inside the cell, and this will be coming under antiporter system. So this is very, very important regarding your NEET. One, you have to remember the sodium potassium pump is an active pump. It is also called a sodium potassium ATPase, and this will be coming under primary active transport. As it is operated with 2 molecules, it is a co-transport system. In co-transport again, the movement of 2 molecules is present and these 2 molecules are operated in opposite direction. We call it as antiporter. As it is primary active transport, the ATP hydrolysis is generally required here. So with the help of ATP, 3 sodium ions are exported out and 2 potassium ions are taken inside. Okay, so this is how the sodium potassium pump will be working. This is present on all the membranes. So this is regarding your primary active transport. So this is one example, children. This is very, very important example regarding your NEET. So you have to remember all these points. And coming to the secondary active transport. So in secondary active transport, I have given 2 examples. You can see this here, children. So this is secondary active transport, and in this, so this is 2 secondary active transport. In this we have 2 examples. One I have given glucose sodium co-transporter, and another I have given sodium chloride co-transporter. So one I have given mainly taken as an example that is present on the small intestine, and another example I have taken which is present on the kidney cells. Okay, so first I will be discussing with the sodium glucose co-transporter. Okay, so here it is secondary active transport. So what is the most important thing first we have to discuss? It is regarding the energy present in the system. So for the movement of molecules against their concentration gradient, so the major important thing here is the energy required. So here I have given these red spots, which means it even requires energy, but the energy, it is not coming from ATP, that you have to remember. So here even I have mentioned as ATP, but here I have not mentioned anything, which means energy is present, energy is required definitely, but this energy, it is created by the gradient. The energy which is derived is mainly by the gradient of one of the molecule operated in the co-transport system. So let us see which molecule it is mainly helping here. So in the case of sodium glucose co-transporter, so this one, which is mainly present on the small intestine. So this is present even on the all cells, but mainly more of these proteins are mainly present on the small intestine. Then what is the significance of this glucose sodium transporter? So name itself indicates the glucose here, it is mainly taken inside the cell cytoplasm along with the sodium. So again these two molecules are, you can say, the companion molecules, they have obligate relationship. And which molecule is sourcing the energy, that is important. In the case of sodium potassium pump, which we have discussed before, both the molecules are moving in uphill manner, the potassium as well as the sodium, but the energy, it is not sourced by any of the molecule, it is sourced by the ATP, which is the cellular currency. But in the case of secondary active transport system, ATP, it is not involved. Then any one of the molecule present in these two, that will be supporting the other molecule for its movement. Then can you tell me which molecule can support? I have told you the importance of sodium in the extracellular region. And this sodium, now you can see, and hence I have given the same example, children. I have told you that for the maintenance of for the uptake of the glucose, which is very essential for all the cells, the glucose will be mainly transported into the cell along with the sodium ion. The glucose, it is not absorbed directly, it needs a companion, and that companion is the sodium ion. Okay, so here the sodium ion, it is operated, sodium ion, it is mainly creating a gradient for the glucose molecule. So which is providing energy here? Not the glucose, the sodium ion. So which is moving downhill, which is moving uphill, that you have to remember. Why? Because we all know the downhill movement will always create the energy, and that energy will be utilized by the other molecule, and in this case the sodium will move downhill and the glucose will be moving against the gradient. Okay, so let us see how this will happen. Before that, I will tell you the significance here. Generally in the small intestine, what will happen, that we will see. So generally what will happen on the small intestine? So in the small intestine, we all know the small intestine, the wall of the small intestine, it is lined by many epithelial cells, and these epithelial cells are called as enterocytes. So let us say this is lumen. This is the hollow cavity. The space which is present in the small intestine is called as the lumen, empty space. And this is the wall of the small intestine, and on this wall what cells we have? Epithelial cells, and these modified epithelial cells with microvilli, these are known as the enterocytes. And other side we have the, let us say, the blood capillaries are present. What will happen here? So generally these transporters, the glucose sodium transporters, are present on the enterocytes. Okay, so here we have the sodium glucose transporter. So what is the significance here? So inside the lumen, the digested food, mainly in the small intestine, the digested food, it will be absorbed and it is mainly releasing different type of substances like amino acids, and the glucose sugars are rapidly released in this particular area. And these available sugars, mainly with the glucose we are discussing. So the glucose, when it is released in the lumen, when it is present, it is quickly absorbed into the enterocytes or the epithelial cell, and later they are transported to the blood capillaries that will be mixed with the blood. So this is how the glucose uptake will be present in the small intestine. Now let us see in this case, the lumen, it will be having which type of concentration here? Even though it is having the low concentration, these enterocytes are constantly absorbing this glucose, means it is against the concentration gradient. So which is moving uphill, children? Glucose. So glucose, even though it is present in less concentration in the lumen, it is constantly absorbed into the enterocyte cells. Okay, so as it is required by the cells, they will be taking in this glucose, a minimum amount, even if it is present, that is quickly absorbed into the enterocyte cells. And here you can have the more concentration, even the glucose is more inside the enterocyte cells, it is in turn supplied to the blood constantly, so this movement is continuous. The small amount of glucose released in the lumen is constantly being imported into the enterocyte, later into the blood. So this is how it will happen. So in this case, if you observe the glucose entry inside the enterocyte cell, this is an uphill movement, right? From low to high concentration, even it is high, it is absorbing the glucose, so it is an uphill movement. And what about the sodium here? Sodium is extracellular. In the extracellular region, it is always more. I have told you the sodium-potassium pump, it is mainly present to maintain this gradient, to maintain the sodium ion concentration. So what is present? The sodium ion concentration is high. Okay. So in the extracellular region, the sodium ion is more, and in this case, the glucose, let us say, it is less. Then what will happen? The sodium, it is moving from high to low. It is low in the case of cell cytoplasm, definitely. So this concentration, the sodium, it is moving in a downhill manner. Okay? And this glucose, it is moving in a uphill manner. So in this case, both the molecules, as they are moving in the same direction, again this is called as a symport, right? So what it is known as symport. Clear with this, children? So mainly one is showing downhill movement and other is showing uphill movement, but they are moving in the same direction. We call this co-transport as symport. So that is what I have mentioned here. In the small intestine, energy is required. Energy is provided by the sodium gradient for the movement of glucose. And why it is happening, what is the significance, that I have discussed here. And in this, I have written the co-transporter. It is known as the symporter. Okay. So next we'll be discussing with this is regarding your sodium glucose co-transporter. And again, I have given another example to make you understand this concept here. So we have another example, that is sodium chloride co-transporter. This is a secondary active, another example for secondary active transport, and the example is sodium chloride co-transporter, and where it is seen mainly on the kidney cells. It is again present on all the cells, but mainly, essentially, these proteins are more on the kidney cells. And what is this transporter called as? Sodium chloride co-transporter. Again, why? Because two molecules are operating here together. We call it as a co-transporter. And in this, let us see the significance first. It is also a co-transporter system, and mainly this is operated on the kidney cells. So here in the system, again the same thing which we have discussed with the glucose sodium transport. In this case, mainly the significance of the sodium chloride co-transporter is to reabsorb this ions from the urine. So in the urine, while processing this urine, the reabsorption of excessive ions present in the urine, it is mainly done by the kidney cells. And in this reabsorption method, mainly the sodium chloride pump here it is operating. And the sodium chloride ions, these are also showing the movement from the extracellular region. The kidney, it is reabsorbing it from the urine, and it is later transported to the blood capillaries to mix inside the blood. Why? Because in order to maintain the blood pressure, mainly these channels, these transporters are operated for the reabsorption of the ions from the urine. The system, this co-transporter is mainly operated. Okay. So significance here I am writing. Right. Now let us see how the movement it is taking place here. So in this case, even the chloride ion, it will be supported by the sodium ion. So one ion has to be supported. This is a secondary active transport. So in the system, ATP will not be present. So one of the molecule it is being benefited with the other molecule. And which molecule is the driving molecule and which is the driven molecule, that we will see. So here the system, it will be mainly operated with the help of the gradient created by the sodium. Means tell me what type of concentration will be present. The sodium ion concentration, it will be high, and here the sodium ion concentration inside the kidney cells, it will be low. So this moment is the downhill. So mainly the extracellular sodium is always high and it will lead to the downhill movement, the same thing which I have explained there. And the movement here is the downhill movement. And the chloride ion concentration, it is less here, but still it has to be absorbed. Any chloride ions or ions which are present in the urine excessively, they will be absorbed by the kidney cells based upon their conditions there. So let us say this is less and it is more inside the kidney cells. Then what will happen? The chloride ion, it will be shifting or it will be moving into the cell in a uphill manner. Okay, so one is downhill and one is uphill, and this uphill movement, it is mainly coordinated and it is supported by the downhill movement, which is creating a gradient in the sodium ion concentration. Okay, so again the same story, the sodium ion gradient will help in the movement of the chloride ion into the kidney cells, and later these are reabsorbed and mixed with the and transported to the blood capillaries. Okay, so this is how the sodium chloride co-transporter will be working. And if you observe the direction here, the movement of sodium and chloride both are taking place in the same direction. Again, we call it as a symporter. So clear with this, children? So one we have discussed with the antiporter, that is in primary active transport. I have taken example of an antiporter, that is sodium potassium pump, and for the secondary active transport I have taken two examples. One is of the sodium chloride co-transporter and another is of the glucose sodium co-transporter. Both are the examples of symporters here. So all these three are the co-transporters. So you should not be confused, children. Even in the case of the primary active transport, we have another transport, that is the uniport system. Only single molecule can be allowed through the membrane, which is known as uniporter. I have told you before. Okay, so in primary active transport we have co-transport as well as the uniport, but in the case of secondary active transport it is purely co-transport. Why? Because one of the molecule, it has to be benefited or it needs support of other molecules, so definitely it requires a co-transport system. And another important thing, co-transport is not always the two molecules, even three molecules will be, more than two molecules will be operated in this system. Okay, so this is regarding your primary active transport and the secondary active transport. And next we'll be discussing with the group translocation. So group translocation is a mechanism. It is also a part of active transport. It requires energy, ATP. Directly it requires ATP, just like the primary active transport, but mainly it is associated with the enzyme system. Enzymes are mainly participated here. So proteins, along with the protein molecules, some enzymes are attached and they will form a multi-subunit complexes. So many molecules, many enzymes and proteins, they bind together to form a complex, what you can say multi-subunit complex, which mainly help in movement of molecule. And one such example I have given here. In the movement of glucose into the cell, this group translocation is taking place. And the major important point in group translocation is that here the molecule which is passed into the cell, it is quickly modified by the enzymes present in the subunits. So here I have told you the enzymes are also present along with this protein pump, and these enzymes, what they will do, they will modify the substrate. So whatever substrate it will pick inside, what they will do immediately, they will modify this substrate. And here I will be explaining this with one example. We all know during glycolysis, once the glucose it is entering into the cell, it is modified into glucose 6-phosphate, and this is mainly done with the mechanism of group translocation. So this group of molecules, they will be importing the glucose inside the cell, and once the glucose it is entering into the cell, it is modified or it is attached to the phosphate and it is converted into glucose 6-phosphate, so that the glucose will not travel back or escape back out of the cell. So this is one kind of mechanism that is seen on the membranes. Okay, so these group translocation mechanism is the one where multi-subunit complexes are involved, and these are mainly the enzyme complexes, and the major important function is that they will modify the substrate. So many molecules we have seen here, but the substrate molecules, they are same when they are present outside, they will be same, and when they enter inside, there is no difference or no change in their conformation. But in the case of group translocation, the substrate which is entering into the cell will be modified. This is the major important point you have to remember. And what example I have given? Glucose, it is modified into glucose 6-phosphate. Okay, this is regarding the group translocation. So glucose will enter inside the cell only with the expense or at the cost of this ATP. Sometimes glucose along with the ATP hydrolysis, only the molecule will be moving inside, that will be coming under active transport, and sometimes these glucose molecules are mainly modified. And why we are always discussing with this glucose? Because it is such a important energy fuel inside the living organisms, the most simplest sugar form. So any carbohydrate which is entered into the cell, it has to be modified and converted into the glucose. Glucose is the major absorbable form of the energy, so it is giving instant energy, and it is the whenever you take glucose inside directly, then only it will be giving instant energy. But if you take any kind of sugar, other sugar, it has to be converted into glucose. Okay, so we have many channels for this glucose. For entry of the glucose into the cell, all the membranes will be having this glucose channel. Passively, actively, in any way this can enter into the cell, and it will be utilized in various conditions. Okay, so this is regarding the active transport.